Hello, everyone. Our lesson for today is about environmental factors affecting microbial growth. In this lesson, you will understand how the different environmental factors affect the growth of microorganisms. You'll also learn the different classification of microorganisms based on their pH, temperature, oxygen, and pressure requirements. At the end of this lesson, you will be able to identify microorganisms based on their pH, temperature, and oxygen and pressure requirements, and describe the mechanisms of how microorganisms combat environmental stress. So the motivation question for this is, do environmental parameters affect microbial growth? I hope you will be able to answer that one as we finish our lesson for today. So the environmental factors that are important that is affecting microbial growth are pH, oxygen, temperature, and pressure. So tolerance for microorganisms have tolerance for variation of 2 to 3 pH units. Now there are three groups of microorganisms based on their pH requirements. There are those that we call acidophiles, neutrophiles, and alkaliphiles. Acidophiles are those that could tolerate pH 0 to 5, meaning these are very acidic environment. Neutrophiles are those that could tolerate a pH range of 5 to 8, and alkaliphiles are those that could tolerate a basic pH from 9 to 11. Now, usually, microorganisms have different metabolic activities that could result to a media that is or a cellular environment that is acidic or basic. So, say for example, there are those that ferment glucose and produce lactic acid. So, in this case, the pH will drop. So, the cell will become acidic. Or in some cases, some microorganisms could ferment amino acids, producing ammonia, and so the pH will rise because this is a basic substance. However, they could combat or they could um, avoid influx in pH or sudden increase or decrease in pH by buffering with KH2 potassium phosphate and KH2HPO4. So these curves here show the approximate pH ranges for the growth of the different classes of a pH-specific prokaryote. So each of the curve have an optimal pH and extreme values at which growth is much reduced. So say for example, for acidophiles, the growth rate is at its highest at in between 3 to 4 pH. For neutrophiles, the highest is at 7, and for alkaliphiles, the highest is in between 9 and 10 pH. So when cells are placed in pH conditions that are below the optimum, below their optimum, this is the optimum, so below that, the protons can enter the cell and lower the internal pH to lethal level, so they may die. Microbes, they can prevent the unwanted influx of protons by exchanging intracellular potassium for intracellular protons or hydrogen ions when the internal pH becomes too low. In some cases, under extremely alkaline conditions, the cells can use a sodium proton antiporter to bring protons into the cell in exchange for expelling sodium. So this is the illustration that shows how a microorganism could avoid sudden influx of these protons. So again, in in a strongly acidic, say for example, pH 2 environment, the enzyme called amino acid decarboxylases will drain the protons from the cell. Okay, So this is an enzyme amino acid carboxylase. It will remove proton, no, from the cell. Under alkaline stress like pH 9, the sodium proton antiport system scavenge protons from the environment. So under alkaline conditions, so meaning the pH becomes uh, basic, no? The cell becomes basic. So what they do, they release sodium and they take in protons from the environment. Okay, so under slightly acidic conditions like pH 5, cells use a potassium proton antiport system to remove internal protons. So in this case, if there is a higher concentration of protons here becoming internal pH more acidic, so they would remove this, the protons, and take in potassium. So that the main purpose here is just to remain, no, the ion concentrations still the same, but the pH is changing. Okay, another factor, environmental factor, is the oxygen. So many microorganisms use oxygen as a terminal electron acceptor in a process called aerobic respiration. So when we go to metabolism, we will understand how the proton motive force works. So in this case, oxygen is needed in order for the proton motive force to work. The proton motive force is the one that is found in the electron transport system, now together with the electron transport system, to produce energy from the electrons uh, that are donated by high energized electron carrier molecules. Okay, so when you test for the oxygen requirements of microorganisms, others will use the thioglycolate broth. So this contains a reducing agent and provides aerobic and anaerobic conditions. Okay, so there are different oxygen requirements of wala dire. Okay, but there are different groups of microorganisms now according to their oxygen requirements. So the aerobes, the anaerobes, the facultative the facultative aerobes, and the microaerophiles, and the aerotolerant and aerobic microorganisms. So in the text discussion, you will find the different uh, definition of these different groups of microorganisms. So when you culture them in a thioglycolate broth, if you see that the um, microorganism has grown more on the bottom of the test tube, then you could say that the microorganism is probably anaerobic, meaning they don't need oxygen for their growth. But if microorganisms grows at the top layer of the broth, then you could say that because this is an oxic environment, meaning this has oxygen in it, so you could say that the microorganisms could be aerobic, or they need oxygen for their growth. If they are facultative, meaning they could tolerate the presence of oxygen, or they could live with or without oxygen. So facultative aerobes, microaerophiles, so they need just a little amount of oxygen for their growth, and aerotolerant, so they are anaerobic microorganisms, but they could tolerate the presence of oxygen. Okay, now oxygen could be toxic, for the microorganisms, and if they are in a different forms. So these toxic oxygen are what we call the reactive oxygen species, or the ROS. So they cause oxidative stress, like damage to membranes, to proteins, as well as to DNA. So what are these different ROS? You have the superoxide, the hydrogen peroxide, and the hydroxyl radical. So how are these ROS formed? So first, when an oxygen no, is auto-oxidized by flavine adenine dinucleotide. Okay, it will take in an extra electron. Okay, so it will be oxidized. So it will produce a superoxide radical union. So it has already a negative charge, meaning no, it has gained an electron. And then this superoxide plus a proton will give you, through the catalysis of superoxide dismutase, will produce another ROS called hydrogen peroxide. Now, if the hydrogen peroxide undergoes Fenton reaction, now it will be broken down into hydroxide and hydroxyl, so it will produce hydroxyl radical. So you have now the formation of three reactive oxygen species, or the ROS. So you have the superoxide radical union, the hydrogen peroxide, and the hydroxyl radical. Okay. However, the cells of microorganisms and even humans have developed ways on how to combat the presence of uh, hydrogen peroxide. So, for example, they have an enzyme called catalase and peroxidase. So, a hydrogen peroxide could be catalyzed by a catalyst to produce water and oxygen, while with action of peroxidase, hydrogen peroxides could be converted to water and nicotinamide adenine no dinucleotide. Okay, so this is another representation of how the ROS are detoxified by an enzyme called the catalase and peroxidase, as well as superoxide dismutase. Another environmental requirement, is the now let us talk about temperature. So the growth of microorganisms is directly uh, affected by the temperature. So in terms of the microbial growth rate, there are three cardinal temperatures that are necessary. These are defined as the minimum, optimum, and maximum temperatures. So minimum temperature is when the microorganisms show membrane gelling and the transport processes are so slow that growth cannot occur. Okay. At the optimum temperature, this is when the enzymatic reactions that are occurring at maximal passable rate, while the maximum temperature is when is a temperature wherein the microorganisms protein are denaturated and there is a collapse of the cytoplasmic membrane and will cause thermolysis. Temperature is a major environmental factors that controls microbial growth. Now we could classify four general types of microorganisms based on their temperature requirement. There are those what we call psychrophiles, mesophiles, thermophiles, and hyperthermophiles. Psychrophiles generally tolerate 0 to 20 degrees Celsius. However, they are further subdivided into two: obligate psychrophile and facultative psychrophile. Obligate psychrophile are those that cannot grow above 20 degrees Celsius, while facultative psychrophiles are those that could tolerate up to 22 degrees Celsius temperature. Mesophiles, on the other hand, are those that thrive well in 15 degrees Celsius to 45 degrees Celsius temperatures. Majority of these mesophilic microorganisms are culturable bacteria like soil bacteria, and all of them are pathogens because this is the temperature that is conducive for the growth of microorganisms inside the body. Thermophiles, on the other hand, are those who can tolerate 40 degrees Celsius, or those that live well at 40 degrees Celsius to 80 degrees Celsius. So these environment are mostly the compost, hot springs, and volcanic areas. Hyperthermophiles, on the other hand, hyperthermophiles now are those microorganisms that thrive well in environment with temperature 65 degrees Celsius up to 121 degrees Celsius. So these are usually also environment with high pressure. So these examples of these are the volcanic areas, not the sea bottom or the hydrothermal vents. Okay, so but above 90 degrees Celsius, so many bacteria are hyperthermophiles, but those microorganisms that could thrive well above 90 degrees Celsius are archaea only, no, not bacteria. Okay, so again, this is a representation of the temperature requirement for microorganisms, and you will find here the maximum temperature, optimum temperature, and maximum temperature. So minimum temperature is the temperature below which enables or which causes growth to cease, or lowest temperature at which microbes will grow. Optimum temperature is the temperature at which its growth rate is the fastest, while maximum temperature is the temperature above which growth ceases, or highest temperature at which microbes will grow. Okay, so these are the definition of these different cardinal temperatures. So how do we classify? Again, the classification of microorganisms by temperature requirements: psychrophile, mesophile, thermophiles, and hyperthermophiles. Okay, so these are examples of the different cardinal temperature requirement for non-photosynthetic prokaryotes and photosynthetic bacteria. Okay, these are the upper temperature limits, no, the optimum temperatures for if you compare the microorganisms to the higher forms of organisms. Another environmental factors affecting microbial growth is the pressure. Okay, so there are microorganisms that we call the barophiles or the piezophiles. So these are those that are adapted to grow at very high pressure, so up to 1000 atmosphere or 101 millipascal or 14,000 psi. There are also those that grow well over the range of 1 to 50, but their growth fails off thereafter. So they're called baro tolerant, no, microorganisms. You note that many biophiles are also psychrophiles, okay, because the average temperature at the ocean floor is 2 degrees Celsius. Okay, there are also there are also hyperthermophiles that are biophiles. If you look in, because higher temperature also could also lead to higher pressure. Okay, so pressure is also affected by osmosis or the influx of water. So osmophiles are those organisms that thrive in high solute concentration. Osmotolerant organisms which tolerate high solute concentration. Halophiles, organisms that thrive in high salt. Halotolerant, they could just tolerate high salt. Okay, and biophiles and barotolerant. Okay, you have here the different water activity of several substances. So if you compare that to the water activity that is for microorganisms, so you can compare. Halophiles are related organisms. So in nature, osmotic effects are of interest, mainly in habitats with high salt environments that have reduced water availability. Now, halophiles, they have evolved to grow best at reduced water potential, and some, like halophiles, extreme halophiles like Halobacterium and Donalera, even require high levels of salts for growth. Halotolerant, they can tolerate some reduction in the water activity of their environment, but generally grow best in the absence of the added solute. They're also what we call the xerophiles, so these are able to grow in very dry environments. Okay, so to summarize, our lesson, so I hope you will be able to answer these questions. What are the different types of microorganisms based on their pH, temperature, oxygen, and pressure requirements? And how do microorganisms combat the ill effects of environmental stress? Okay, so that ends our lesson for today. If you have any questions, you just contact me or your respective lecture instructor. My email address is here. I also would like to thank Miss Maria Shalita Estersal for the help in making the slides. Okay, thank you very much for your attention and see you again in our next lesson.